and I'd like to congratulate. Just a minute. I think it's recording now. I will repeat. Thank you very much. First of all, thank you very much, Miguel. I think this is a pleasure for me to be here. And I'd like to congratulate you for this amazing initiative, which is connecting high top level bioinformaticians from different regions of the world with our community here, not only in Brazil, but also in the whole Latin America. As we already discussed in other opportunities, this kind of event of seminar series is very important to spread the word on the bioinformatics. And now I'd like to introduce you guys to Dr. Christophe Dubessy. I hope I pronounced correctly your name, Christophe. And
yeah,
he's amazing. He's an associate professor at the University of Rouen, Normandy, in France. He has a wonderful work studying the secretion of neurohormones and its regulation by microRNAs. And today he will talk about a very nice tool that he developed called Mirabel, which was designed to perform the efficient prediction of microRNA targets. And he will also talk about the application of this web tool in studying hormone hypersecretion in neuroendocrine cancer. Dr. Christophe, thank you very much to be here, and I'm eager to hear your talk.
Okay.
Welcome.
Okay. Thank you, Vinicius. I'm going to switch on my presentation. Here. Okay. Is it okay for you? Do you see my presentation?
Yes, it's working perfectly.
Okay, and also my mouse.
Yes, we can see the mouse. Let's see when you full screen mode. Yeah, it's perfect.
Okay.
We can see the mouse and the full screen presentation.
Okay, this is the full screen. Okay. Okay, it's a pleasure for me to be here. First, I would like to thank Professor Ortega for inviting me to present my work some months ago. It's a great honor for me to give this lecture for an audience of bioinformaticians. I would like to specify right away that I am a molecular biologist and a cell biologist, and not a bioinformatician. And I am interested in bioinformatics approaches for my research purposes. I will explain during my presentation. So, as you say, Vinicius, I am a senior lecturer at the University of Rouen, Normandy in France. And I am interested in neuroendocrine tumors, and more particularly, the role of microRNAs in these cancers. Today, I will talk about the improvement of bioinformatics predictions of microRNA targets by rank aggregation. And in the second part, I will show you how we can use this prediction tool to apply it to pheochromocytoma, which is a new endocrine tumor. Okay. Some very brief reminders about microRNAs in vegetarian animals. My mouse, where is it? Yes, it's okay. MicroRNAs are genes, as you know, whose primary transcript here is maturated in 2 steps, by drosha in the nucleus and by dicer in the cytosol. The microRNAs form a duplex RNA of about 20 nucleotides, 22 nucleotides. The most stable strand is taken over by an agoprotein to form the myrisk complex, which will replay its target mRNAs. but probably you know this basic molecular biology information. To do this, the microRNA will bind imperfectly in the 3' UTR of the mRNA. In fact, the pairing takes place at a small place. You can see here, it's a really small sequence of 6 to 8 nucleotides called the seed sequence. The rest of the microRNA sequence reinforces this binding here, for example. The Mirix complex acts mainly by destabilizing the mRNA. It induces deadenylation and decaping also of UTR. Mirix can also inhibit the translation, but this is rarer. It's also important to know that direct mRNA cleavage is quite rare in animals, in bilaterian animals, unlike in plants. An important point is that microRNA can regulate up to several hundred target mRNAs, probably around 400, but it's not really clear. But this number depends mostly on the cellular context. Conversely, one mRNA can be regulated by around 4 to 6 microRNAs at the maximum level. Thus, microRNA seems to be involved in all cellular processes, such as development, differentiation, apoptosis, metabolism, proliferation, cell proliferation, and so on. Another information about microRNAs, they can therefore be considered as an additional level in the control of gene expression. Thus, in a cell, the level of gene expression, as you probably know, here in blue, for different genes here, is controlled by transcription factors and by level of chromatin condensation. The microRNAs repress this level of transcription. And the most binding side they are, the greater the repression, as you can see here. Okay. In the end, microRNAs constitute a complex network that finely regulates gene expression and therefore the activity of all cellular processes. The question that arises, therefore, how to identify the target mRNAs of microRNAs. A first approach is based on heuristics methods here. In this case, you have several analyzable elements that characterize the interaction between a microRNA and its target. And these analyzable elements can be used. For example, the seed sequence, the phylogenetic conservation here, or several thermodynamic parameters between for the microRNA or for the 3' UTR. Another way to identify microRNA targets is to use machine learning methods based on experimental gene or protein expression data here. Analyses are done with low throughput methods, such as Western blot, luciferase reporter, but also with high throughput methods, such as microarray or sequencing technologies, also proteomic analysis for PCR, for example. These two types of approach are sometimes combined, And in the end, we recently counted that there are currently nearly 200 prediction tools available on internet. And I just want to say that these methods are described in a paper that had just been accepted for publications 2 days ago, very recent. This paper will be available soon in a computational and structural biotechnology journal. I have represented here 60 of these prediction tools, because I just said before that there are around 200 target prediction tools available. And here I have represented 60 of these prediction tools on this word cloud based on the number of citations. Some of them are highly cited, like target scan. Probably some of you know target scan. Miranda here, RNA22, MirDB, Victor, Peter, Diana, and so on. Overall, bioinformatic tools are interesting because they allow a quick and a cheap first selection of target mRNAs. So it is very useful for that. Each of these tools has its strengths and weaknesses. Unfortunately, they are not always updated, and their performances are often poor. For example, some of these tools are widely used, but have not been updated for more than 10 years. 10 years. It's really hard for a prediction tool concerning target mRNA. And other tools are dedicated to only one species, such as Drosophila or C. elegans. And in this case, it's not possible to use these prediction tools for rats, human, or mice, for example. In fact, the bioinformatic prediction of MicroRNA targets remain a great challenge. Indeed, when we search for the targets of microRNA with several tools, we obtain quite different results, both in terms of predicted target mRNAs and in terms of number of predicted targets. Therefore, we proposed some years ago a solution that consists in aggregating the results list of several prediction software and reordering these results according to their ranks, their respective ranks. And this is the main idea of Mirabel I will present you now. So we developed a new prediction tool called Mirabel. Here, this is Mirabel, its logo. We developed this tool in collaboration with Professor Thierry Lecroc from the LITIS lab here in Rouen. And this tool aggregates the result, the prediction list of 4 prediction tools: Miranda, PITA, SVMicro, and also Target Scan. It also uses predictions located in the coding sequence here and in the 5' UTR. And this prediction comes from Mirwalk, the Mirwalk database. Mirabel also integrates experimentally validated interactions from 2 databases, MirTarbase and MirRecords. And we also include the KEG signaling pathway. Since the annotation of microRNAs has changed over the year, a tool was developed to convert the names of microRNA queries in the latest version used by Mirabel. Mirabel is the reference database of microRNAs. And this tool is also accessible if you want to convert the name of your microRNA of interest. The tool is included in Mirabel. In order to standardize gene names also from different tools, these names were converted to the NCBI gene ID. All this information was stored in a database designed with MySQL. And in the end, Mirabel, the actual version of Mirabel, includes nearly 15 million interactions, including around 350,000 experimentally validated. Almost all human microRNAs and human genes, and 275 C19 pathway from KEG. The first question is which rank aggregation method should we use to construct Mirabel? In fact, we tested 5 methods using our language: the mean, the robust rank aggregation, the geometric mean, the median, and the minimum. And all of these methods are included in a package for R. This is the package. The name of this package is robust rank aggregation. And to do this, we use 10 random dataset of 50,000 experimentally validated microRNA mRNA interactions retrieved from Mirabel and Mirabel. And this prediction are very information are very important to build the database of Mirabel. And it appears that the mean of the rank here achieves the best performance in terms of sensitivity. The sensitivity is here, the true positive rate, and the specificity corresponding to the false positive rate, as can be seen on this ROC curve. ROC means receiver operating characteristic. We also perform other analysis, such as precision recall and F-score analysis, which show that the mean of the rank is always the best aggregation method to aggregate the prediction of Miranda, PITA, SV micro, and target scan. What's about the performance evaluation of Mirabel? It appears that R2 is significantly more sensitive and more specific than databases that are aggregated, as can be seen on this ROC curve. In fact, we tested here the prediction, is the ROC curve here, the efficacy of the prediction for Mirabel and also for Miranda, PITA, SV micro, and target scan. And Mirabel is always the best. When compared to other databases, again, Mirabel is significantly better than than 4 other databases. Here we used MBStar, MirMap, MirWalk, and XPRAG. We also highlighted the fact that the performance improvement is mostly on the top of the prediction, among the most likely target mRNAs. So it was a great result for us because it's possible to, we showed that it's possible to aggregate the prediction from different prediction tools to improve the prediction efficacy. We have also built a website for researchers to, in fact, just to easily access our predictions. It is hosted at the University of Rouen, Normandy. You have the URL here. The main idea is really to have a free and an easy to use tool. It's really important for us. However, I had contacts with several users in the last months, and the ease of use is really a criterion of choice for biologists, especially those who are not familiar with coding, for example, or running script, or the use of a bioconductor, for example. As you can see on this screenshot, Mirabel accepts MicroRNAs, just here, MicroRNAs, genes or pathway as input. In fact, you can also enter a list with several microRNAs or genes. At the left, you have an example when it's possible to, where it's possible to type the name of several microRNAs in this example. And on the right, you have the same possibility with the genes. The results are pre-calculated and displayed in few seconds, generally between 5 to 10 seconds. Yes, it takes around 5 to 10 seconds to obtain the predictions. For example, when you enter a microRNA, here this is an example with miR16 in human, you get a list of target mRNAs, which are ranked according to the Mirabel score. As you can see on this screenshot, multiple queries are allowed in order to identify common prediction results for both microRNA and gene. And the smaller the score, the stronger the prediction with Mirabel. The full name of the gene is displayed when you move over the prediction, possible here, sorry, in the screenshot. In the columns on the right, you can see the ranks for each of the databases we use. Again, PETA, Miranda, Micro, and TargetScan. For example, for MIR16, the best gene is FAM7OA. Yeah? The Mirabel score is very low. This is very good. And the score, the rank for this gene in PETA, Miranda, and SV Micro and TargetScan is displayed just here. We have also integrated further information regarding the experimental validation of the interaction as well as the possible binding of the microRNA in the 5' UTR and within the coding sequence, CES coding sequence. Okay, and again for this gene predicted as an mRNA target for miR16, you can see that this interaction has been experimented, validated. For the prediction made by Mirabel, it is also possible to predict the signaling pathway potentially impacted by this microRNA. Again, this is an example and with miR16 again, and you have the cake pathway of coming from retrieved from quick cake, which are displayed here. And for example, miR16 impact 65% of this pathway, diglutamine and diglutamate metabolism. And you have the percentage with the number of targets and the total number of genes and the number of experimentally validated targets here. So it's easy to understand the potential impact of microRNA and microRNA on different signaling pathway using Mirabel. What next now? In fact, I have had the opportunity to present Mirabel at conferences, and the tool has been well received before the publication and after the publication. This tool has been well received by researchers working on mRNA target prediction. And this is confirmed by the various impact measurement since publication. For example, the number of downloads or citations are constantly increasing since the publication in January 2020. We also had a contact with several users who made me suggestions to improve the tool, and I appreciate this contact. This fact encouraged us to continue this work, and we are now in the process of defining how to improve Mirabel in order to develop a second version of Mirabel. Currently, we have several lines of development. For example, we will integrate other databases to improve predictions. But the main challenge is to know which ones to integrate and how to combine them in an optimal way. Preliminary tests show that there is still room for improvement. As you can see on this ROC curve, recently we showed that it's possible to increase the sensitivity and the specificity of Mirabel using other databases. But this is only a preliminary result. On the other hand, we would like to consider 4 interaction sites between microRNAs and mRNA. And the current version of Mirabel only includes the most likely interaction sites. So we want to improve this fact. Another axis of development consists in integrating the predictions of other species than human beings. Effectively, Mirabel works only with a human being, actually. And we plan to integrate rat, mice, zebrafish, C. elegans, and flies, Drosera melanogaster, because these species are widely used in microRNA research. Finally, we will improve the interface, the web interface, by considering the user's remark. I would now like to show you how Mirabel can help biologists to progress in their research. I will take the case of the research we're currently doing in my lab. It aims to highlight the role of microRNAs in the control of hormone secretion, neurohormone, in neuroendocrine cancer. In fact, I'm working more particularly on pheochromocytoma. Pheochromocytoma, which develops from the adrenal gland, which is here. The adrenal gland is just here, and this pheochromocytoma arise from chromatin cells of the adrenal medulla. It is a rare tumor because its incidence is about 1 case per 100,000 people per year. Okay? And this tumor has 2 major characteristics. There are about 35 to 40% of familial forms, which is very, very high. Indeed, about 20 driver genes associated with pheochromocytoma are known, actually. But the other characteristics that interest me more particularly is that pheochromocytoma secretes a high quantity of hormones neurohormones in the blood, in particular catecholamines, such as dopamine, adrenaline, and noradrenaline. This hypersecretion is a result of a deregulation at the cellular mechanism. Is it okay? Yes, it's okay. Sorry. Yes, it's in fact, this is a deregulation of the cellular mechanism of the secretion. And we have demonstrated this in a work that we have just submitted for publication, and just submitted the first time we submitted this work. And then the catecholamines are produced and stored here in secretory vesicles here that are transported to the plasma membrane. You can see here. Okay. Upon nerve stimulation, these vesicles fuse with the plasma membrane. We have a magnification here. This secretory vesicle releases their content into the bloodstream. This excess of adrenaline and noradrenaline then cause numerous deleterious effects on the cardiovascular and pulmonary systems, which can lead to the death of patients. We hypothesize that microRNA could be involved in this deregulation of secretion observed in pheochromocytoma, and more widely, perhaps in neuroendocrine tumors. In order to identify the microRNAs involved in exocytosis, the secretion process, in fact, we proceeded in several steps. We started by listing the proteins involved in exocytosis, which is the final step of hormone secretion. For this, we relied on information available in several databases, such as GO, Gene Ontology, KEGG, and StringDB also. We manually validated this information through literature review. take us a long time to validate that. And this approach allows us to identify 96 genes involved in catecholamine exocytosis. The relationships between these proteins were represented with the cytoscape software. And the analysis of this network, according to the interactions between proteins, show that is self-centered, as you can see here. Okay. It's just the first step. For the second step, the objective was to identify the microRNAs that regulate the expression of these genes involved in exocytosis. For this, we use Mirabel to perform the reverse prediction. We enter the list of the 96 genes, and Mirabel returned us a list of 221 microRNAs with a high probability of interactions. We then use the OncoMir here, the OncoMir tool, which allows to identify pairs of microRNAs and mRNAs that are inversely co-expressed. And for this purpose, OncoMir exploits the TCGA database, which includes expression data for more than 10,000 tumors and 33 cancer types. This strategy allows us to retain only 186 microRNAs that are inversely co-expressed with their potential targets. All this information was then represented with the cytoscape, and we call this network ExoDB. Hope you can see that. Yes, okay, it's okay for me. Okay, and the name of this network is ExoDB, Exo database. The microRNAs are represented with blue circles, and their size, it's possible to start just here. I hope you can see my mouse. Okay, and their size is proportional to the number of interactions. For more readability, we have organized the network according to the number of interactions for each microRNA. The genes correspond to the white rectangle in the center of the network. Okay. This network has more than 1,000 potential interactions between microRNAs and mRNAs. These interactions are therefore predicted by Mirabel. Those that are experimentally validated are in red. Hope you can see that. For example, this big straight line in red is a predicted experimentally validated interaction. It's indicated here. We can see on this network that 14 microRNAs here, the first 14 microRNAs, have more than 10 potential interactions with the genes. To our knowledge, this is the first interaction network between microRNAs and mRNAs involved in the regulation of exocytosis. The final idea is obviously to identify only the microRNAs and genes involved in the deregulation of secretion in pheochromocytoma. I explained you just at the beginning of my talk. For this purpose, we use two data sets available on Geo that allow us to identify genes here in blue and microRNA here in yellow. that are differentially expressed between normal adrenal gland and the pheochromocytoma. This is just a differential expression analysis. From our previous ExoDB data, we then extracted genes and microRNA involved in exocytosis, which allow us to identify finally 38 genes and 28 microRNAs whose expression is deregulated in pheochromocytoma. And these genes and microRNAs are linked to each other. We use these genes and microRNA to construct a second network, a third network with cytoscape. And this is the result. This can be seen on this network. There is, in fact, a general underexpression of microRNAs in pheochromocytoma, corresponding to the blue circle. When the color is blue, it means underexpressed. You can see here. Thus a general overexpression of their target mRNAs, corresponding to the red square. Okay, so the microRNAs are generally underexpressed in pheochromocytoma, and their corresponding genes, potential target genes, are overexpressed. Interestingly, the genes that are overexpressed here in red are known to be directly involved in the core of the exocytosis machinery. This is the case for the SNAP proteins, SNAP25, syntaxin 1A. For example, you have a syntaxin STX3 here, STXBP1 here, and these genes are really in the core machinery of exocytosis. There are also regulators of snare complexes, such as CYT1, synaptotagin 1, here, CYT1, VAM2, I don't remember where is VAM2, I don't find it. Sorry, I don't remember where is VAM2. Anyway, STXBP1, and there are also calcium sensors such as RIMS S1 or RIMS 3. And this protein, calcium sensor, are very, very important because they bind calcium and they allow the fusion of secretory vesicles with the plasma membrane. The network contains 133, just indicated here, 133 interactions, and of which 25 are experimentally validated in red again. We can also see that this network is self-centered when we consider the prediction of Mirabel and the co-expression data of Oncomer. These results are consistent with an overactivation of secretion in pheochromocytoma. Okay, if you have an overexpression of this protein, it induces an overactivation of secretion. We are particularly interested by this microRNA, MIR34A5P. I will explain you just on the next slide what we done recently with this microRNA. In fact, recently we wanted to experimentally test one of these microRNA, and we choose MIR34A5P, which is one of the most deregulated microRNAs. You can see here. It is one of the most deregulated in ferrochromocytoma. And moreover, it targets 14 genes of the network. For this purpose, we use a cellular test. In fact, we use a rat ferrochromocytoma cell line called PC12. And this PC12 cell line is stably expressed a luminant reporter gene. In fact, this gene is a fusion protein between growth hormone, human growth hormone, and nanoluciferase. It's quite small, but you have here molecular modelization of human GH1 and the nanoluciferase. And this reporter is specifically localized in the secretory vesicles that store hormones, neurohormones. So this fusion protein acts as a reporter, a luminescent reporter, and it allows us to measure the secretion activity. We have shown that these cells are stimulable with potassium chloride, nicotine, or barium. So we also showed that the secretion rates of the reporter and the catecholamines are similar to each other. So it validates, this fact validates our cell line as a good test, a good tool, a good assay to measure secretion activity. We transfected the MIR34A5P into the cell line, and upon stimulation with barium, okay, and we observe a significant decrease here, a significant decrease of about 40% of the secretion over time. This kinetics lasts for 30 minutes. Okay. In blue, you have the negative control. This is the secretion rate of this cell line during 30 minutes. And when we transfect microRNA 34A5P, you can observe this huge decrease in the secretion activity of the cell. And this inhibition is even greater than that caused by the positive control. We have a positive control here. In fact, this is a small interfering RNA directed against syntaxin 1A, which is a key protein involved in exocytosis. And in fact, MIR34A5P is better than our positive control. And interestingly, when the cells were rested after transfection, a significant accumulation of around 66% of the luminescent reporter within the cell was observed. Okay, here you don't have any stimulation. The cells are rested. Okay, were rested. And this results was confirmed by transfecting increasing doses of the microRNA, and again, dose-dependent inhibition upon cell stimulation and an accumulation of the luminescent reporter when the cells are at rest was observed. So this is a great result because we identify microRNA involved in the secretion, the regulation of the secretion in ferrochromocytoma. In conclusion, we use the Mirabel. Here you have Mirabel, and the use of Mirabel for us to identify the microRNAs involved in excess hormone secretion in pheochromocytoma. We see that the regulation of the expression of a set of microRNAs, including miR34A5P, is associated with an overactivation of the secretion pathway. Okay, this is the situation in the normal adrenal medulla and the situation in pheochromocytoma, where the expression of miR34A5P is reduced with other microRNA, and this decreased expression of microRNA lead to an increase of secretion. In fact, this would lead to hypersecretion of hormone, such as catecholamine, which induce deleterious effects in patients. We are currently screening the remaining 27 microRNAs. You remember, we identified 28 microRNAs, and we just test only one, actually, miR34A5P. And we have, again, 27 other microRNAs. And the assays are not yet complete, but our first results show that more than 2/3 of them have the ability to regulate secretion. If this result is confirmed, it validates our bioinformatics approach and the quality of the prediction made by Mirabel. In addition, we will soon measure the effect of these microRNAs on the expression of the 38 target genes we have identified. For this, we are developing a quantitative PCR panel. We're also evaluating the effect of 3 of these microRNA, including miR34A5P, by TIRF microscopy and enterometry. TIRF, I don't know if you ever heard about TIRF. TIRF means total internal reflection fluorescence. And in fact, these two techniques, TIRF and enterometry, these two techniques, allow us to analyze single exocytosis events at the membrane level, and thus better understand how these microRNA exert their effects. Ultimately, we hope to better understand the mechanisms involved in the deregulation of secretory mechanisms in pheochromocytoma and also in neuroendocrine tumors. Finally, I would like to thank the people who are involved in this project, in particular Aurélien Quillet. Aurélien is here. Aurélien Quillet, who did his thesis with me on Mirabel, and Shadi Sahad here, who worked during his master at the very beginning of this project some years ago. I would like to thank also Ines Risa here in blue, who is currently working with me for her thesis on the role of microRNA in the deregulation of secretory pathway in pheochromocytoma. She performed the experiment I showed you on the last slides. And this project would not have been possible without the involvement of Professor Thierry Lecroc here. Thierry Lecroc, who leads a research team in bioinformatics here in Rouen. We also work with several other research teams in France in statistics, mathematics and statistics, and also with doctors from hospital in Bordeaux and in Toulouse in France. I do not forget our sponsors, the INSERM, the University of Rouen Normandy, the region Normandy, and also the Ligue contre le cancer, which is a caritative foundation, very important here in France. thank you for your attention. And to finish, I propose you some view of our beautiful region and of Rouen. You have Rouen just here, the Normandy is here, Paris here, and you have a sightseeing view of the city center of Rouen and the River Seine. Our lab is just here. It's not possible to see our lab is just here. This is a street of Rouen here, street of Middle Ages, very ancient street. Perhaps you know Claude Monet, the famous painter. Claude Monet has painting the cathedral of Rouen. And in Normandy, you have also very beautiful places such as Étretat. This is the cliffs of Étretat. The highland of Mont Saint-Michel, very famous. The harbor of Honfleur, very tiny city. And the tapestry of Bayeux, the first comics in the world. It explains the conquest of England by the Duke of Normandy. And that's all. Any question?
Thank you very much, Christophe, and thank you for the wonderful pictures from Rouen. And congratulations for the talk and for the tool. As far as I know, we had 61 attendees. And
yeah.
I have some comments, but also I would like to ask the audience, if you want to make some questions, please post your questions in the chat and I can read to Dr. Christophe. Some comments, considering the integration with other species would be amazing, and of course would increase even more the impact of Mirabel. And I have two simple questions before I move to the audience questions. How did you select the 4 prediction tools, Miranda, PITA, SVM Micro, and Target Scan? Which were the one you implemented? Which filters did you consider?
Well, I think it's just at the beginning of this project, we wanted to integrate the results of different prediction tools, as I explained, but with possibly with different features. When we began this project some years ago, there were, I don't know exactly, perhaps 100. I don't know exactly how many prediction tools were available, but it was important for us to select tools with different features. For example, some of them use only seed characteristics. Mm-hmm. Other use only thermodynamics characteristics. Some of them include how to say that, include a conservation information, phylogenetic conservation information. Perhaps I can show you this slide. I don't remember where. Yes, it is. No. And I don't know. Do you see my present? Oh no, you can see my presentation.
Yes, we can see.
We see.
Okay. Okay, and some tools use only conservation, other only the context. But so it's important for us to combine these different features, these different characteristics, to increase the performances of the predictions. But recently, more and more tools integrate these different features, heuristic features. For example, target scan. Yeah. Target scan include most of these characteristics, but not all of these characteristics. So in fact, when you merge all the results, it's possible to increase the performances of the tools or of the prediction. So it was our main interest just at the beginning of this project.
Perfect, thank you. Another simple question: is it possible to use the nucleotide sequence of a transcript to run the target prediction?
No.
Oh, no, it's already the computed. Yeah.
No, it's not possible because again, how to say that, one of the main interests when we developed Mirabel was to develop a tool which is easily accessible for biologists. We used who works, for example, on one microRNA or two or three, or a small set of microRNAs. And generally, they say, I have a microRNA, and what are the main targets of my microRNA? And if they use, for example, I can switch. If they use a first tool, they obtain a list. A second tool, they obtain another list of results. A third tool, they obtain a third list. And these lists are quite different. Yeah. So it's just for Mirabel is dedicated for to biologists, basic biologists, who need to work with targets and microRNA. But if they want to manipulate directly the nucleotide sequence of their microRNA, it's not possible because Mirabel is not dedicated to search for the genomes, for example.
Thank you. Yeah, it's a very nice tool to fine-tune the predictions and obtain the targets, and really nice. I have some very nice very nice questions from the audience. Isabella Mamede, she made two questions. I will start with the first one. She says, very interesting work, Professor. I have two questions. The first one, let's start for that. Is Mirabel able to predict microRNA long non-coding RNA interactions or only for messenger RNAs?
Oh, currently Mirabel works only with mRNA, but I didn't mention that during my talk, but no, just here. Yes, we plan to develop this kind of function for the next version of Mirabel, because several researchers asked me exactly the same question: is it possible to predict interaction between microRNA and LNC-RNA? And probably yes, and we plan to introduce this function in the next version of Mirabel. And the development has just begun some weeks ago.
That's amazing, because now it's increasing the evidence and the reports and the associations with disease, the role of some long non-coding RNAs as acting as sequestering the microRNAs, avoiding them to like the compete endogenous RNAs.
Yes, in fact, this is a competition.
Yeah, and
in fact, they act. LNC-RNA can act like sponges, for example, and often we find this these terms in publication. Some of them can act like sponges to regulate the level of microRNAs in the cell.
Another question from Isabella: is it possible to differentiate which mRNA isoforms interact with any specific microRNA? Let's say if a specific mRNA gene code isoform if you have the specific prediction according to isoforms and how does Mirabel works on this?
Yeah. In fact, we introduce and we introduce in Mirabel or microRNAs, human microRNAs, currently known actually, that are present in the Mir-based. In fact, we use the Mir-based database, and if there are different isoforms of microRNAs which are included in Mir-based, in fact, they are included also in Mirabel.
I got it. That's quite important. Also, we work with sponge microRNAs, and we are trying to integrate the different databases with predicted and validated interaction. And this was my student complaining a lot because all the database, they did not differentiate the isoforms that were there. And yes, this is the real life. Sorry, this is what we have in the database, and we only can work with what we have. Thank you. And I have another 2 questions from Moisés Catonio. And he says, great work and great presentation, Professor. I have 2 questions. What were the motivation to choose R as the main language for the tool development? And would Python and all the libraries available serve as an alternative development language for a software of this complexity?
In fact, just at the beginning, Mirabel, it is a long story, a very long story. And just at the beginning of the development of Mirabel, I had worked with a student, Shadi Saad, and Shadi preferred to code with R. And I'm a cell biologist, okay? I'm not a bioinformatician. Now I just began to code some little things with R or with Python. But just at the beginning, I was really naive about that. And Shadi just began to code with R. And our first tests were made some years ago with only 2 databases and with a really small set of microRNA and a really small set of genes. It's just to obtain a proof of concept. We wanted just to know if it works or not. And it's just after that, and Shadi did a really great job, but with a really small set of genes, microRNAs, and databases. And after that, Aurélien Quillet, who did his thesis with me, continue this project, and he continued with R. And that's why actually all the prediction, because the predictions are pre-calculated when you connect to Mirabel. In fact, all predictions are pre-calculated. All the predictions are made with a script coded in R. I agree with you. I think it's probably better now to use Python and the library, but now we just begin the development of the second version of Mirabel, and we plan to change our code and to use Python probably to develop the second version of Mirabel, because it's too difficult to use R to develop all the structure of Mirabel.
Thank you, Christophe. And this is a question that I have tools developed in, not me actually. I program some language, but not all that I will mention here. The students, I have tools in Java, in R, in Python, and Perl, and they have some small fights. No, it would be better to develop in this language, in another language. And I say the best language is the one that you are used to and you are able to implement. I have no prejudice with any language. Actually, I am a Perl developer and R. I already started years ago Python. I said I don't have time to go further on these. I know that it's not
and to learn another language.
Yeah. And Miguel, please, do you want to say something?
Christophe, when I saw the results of sensitivity and specificity, I confess that I was not very excited, because the performance of the individual tools are not that great. That rock curve was not that great. And even assembling them, of course, you had some improvements, but even though the performance of the match microRNA target, I thought it was near to the aleatory, so I was not very excited at this point. But I would like to comment that when you move it to the study case, then actually it seemed to me that you combined the result of those 97 proteins. First, you curated proteins related to a phenomenon, right? So you did a curation. And then later on, you kind of combined the scores of those microRNAs and proteins. There is a combination. You did not convert that to a number, to a score. But when you have this last slide, when you have the microRNA with a big blue circle and etc.
Here?
No, this is scores. When you get to this, actually you can do an assembling of the individual numbers and get a score that would say, well, in my analysis, I selected some 96 genes and 100 something microRNAs. I combined it. They have individual scores. They have expression and increased, lower expression, etc. Actually, in Mirabel 2 or 3, you could try to combine those results to have a score that would say, well, I will investigate this microRNA because it's a big blue circle and has so many connections and etc. And I can tell you that an expert on bioinformatics it's possible to collect the data that you used here in large scale, like to select a network of proteins and to bring the results of expression, etc., and execute this analysis that you did. Because if I understood correctly, after you saw this figure, then you could pick the microRNA to test experimentally, right? So
Yes.
this part of the talk, I think it could reveal to me that there is something there to test. And I think that was what you did, right? When you saw
Yes,
when
yes.
you saw this result, you decided to test something, right?
Yes, in fact, we prefer to use a bioinformatic approach at first, just to select the best candidate. It's just the purpose of this work, this part of the work, just to select the best candidates, because there are too many microRNAs to test. It's not possible to test all the microRNAs experimentally test on the secretion, because the secretion test is has a very low throughput. So it's possible to test some tens of microRNA, but it's not possible to test several thousand microRNA. It's not possible. So that's why we choose to use a bioinformatic approach at first, and to select a subnetwork of protein. Okay, only the subnetwork concerning the exocytosis process in the cells, and to predict the microRNA that regulates this network of proteins. And in fact, it works. It works well, because around 2/3, our last results indicate that 2/3 of our microRNAs, around I don't remember exactly, perhaps around 18 among the 28 microRNAs we selected, 18 of these microRNAs have a really great impact, a really great effect on the secretion. So I think, I'm not sure, but I think that our approach validates this result, in fact, validates our bioinformatic approach using Mirabel and the prediction made by Mirabel. I'm not sure about that, because we have tested only MIR34A5P on the secretion with a large number, using a large number of replicates. But just yesterday, in fact, we obtained, we have actually 4 replicates for the 28 microRNAs. And it's not enough, but we have 4 replicates now. And it works for about 18 microRNAs. So it's just amazing to obtain such rate of success. If we pick microRNA, random microRNA, and we try to test them, it's impossible to obtain around 60 to 70% of success rate. And to confirm that, we choose also negative microRNA. I did not explain that on my talk, during my talk, but we choose several negative microRNA for secretion. In fact, we predict, we predict microRNA with no known protein implicated in the exocytosis pathway. And we tested several of them, 5 of them, and none of this microRNA have an effect on the secretion. So it's really interesting to see that it's possible to use this tool Mirabel, and all the process all the workflow I described to you to select the best candidate to the best candidate for this secretion activity.
Sure, but my point is that okay, Mirabel gave you a list of microRNAs to test that are related to those targets, but the procedure that you did actually got the results. And we look at this figure, we see a big circle with connections, with a blue color. Those things could be converted to a probability, to a score, you know.
No.
Without us looking to the figure, we would numerically pick the 34A5P as a candidate.
For example,
a calculation for the procedure that you do.
For example,
the combination got the results. I think so. Your procedure has to be followed. What you did with the procedure is even more informative than the Mirabel itself, because Mirabel is microRNA target. But when you combine this information in this, then the candidates pop.
Yes, yes, I agree. For example, you talk about MIR 424, okay, the biggest one, okay. For example, for this microRNA, in fact, this microRNA has no effect on secretion. No effect.
Which one was the good one? The blue one.
In fact, one of the best is MIR 34A5P. We have also huge effect of MIR 195-5P here. 497-5P here. We have also 32-5P. Yes, it works very well. 365, one of the best is 365A3P here, which is one of the best. And it's not always, I agree, we have a big blue circle, and when we see that, we can think, oh, it's the best, it's probably one of the best microRNA. No, it's not necessarily the best microRNA. It's one of the best. It's one possibility. In fact, the size represents the number of interactions. As you can see here, this is the number of interactions. And it's not because a microRNA has a great number of interaction that it will have an effect on this cellular process, on the secretion. Perhaps only a small, just a small blue circle with a reduced number of a low number of connection will have a better effect on secretion. And it's hard to say to predict the intensity of effect of this microRNA or of the different microRNA. It's not possible for me to predict that. It's just an information. The number of connections is important.
Okay.
the score, the Mirabel score is important. The level of expression also in pheochromocytoma is also important.
You did not consider in this analysis the number of connections that they have with this network compared to other networks. I mean, are they specific for the exocytosis network? You know, this could be also considered. But I think that the also bioinformatically speaking, you could bring the information about the difference in expression also from for a next tool, right? This information can be put in the analysis automatically. So I would suggest you to try to work on this figure, the way you do this figure, and try to develop a tool that would automatically do this analysis and maybe compare the scores to see if it's important to be dark blue, not less than 5 connections.
Yes,
right?
yes, it's a good
thing like that.
It's really interesting.
I think that your success on the biological case study was relied on the analysis that you did after Mirabel, but this analysis that you did is great to follow. I would follow your procedure if I had a phenomenon to study participation of microRNA. I would not stop at the tool. I would do exactly what you did. This networking, right?
Okay. Okay.
All right, Vinicius.
I was mute. Thank you, Miguel. Do you have any additional questions from the audience, please, guys? Yeah. If not, thank you again, Miguel, for the invitation to be here, the share of this great talk. And thank you very much, Christopher, for this wonderful talk. I'm eager to visit your city and also to make use of the tool. We are working a lot with non-coding RNAs, microRNAs, long non-coding RNAs, and these interactions. We are working on on the sponge mechanism of some long non-coding RNAs and circular RNAs. And I believe your tool will be very useful for our candidate selection. Thank you very much.
Thank you. Thank you for your attention, and thank you for inviting me. It was a really great pleasure to be here. Really, it's nice.
Thank you.
Thanks a lot, Christophe. Great.
Okay. Thank you again. Bye.
Bye-bye.
Bye-bye. Thanks, guys.
Thank you.